Okay, so hi guys, welcome. Good morning, good afternoon, depending on where you are. And I'm Alejandro Vignoni, and I will be giving this second webinar about modeling on behalf of the iGEM measurement committee. So, as usual, the material, these slides, and the scripts that we will be using from MATLAB, there will be accessible to all of you through the Git repository of the measurement committee. There is a link at the end, and in the iGEM webpage, you can also find it if you want. Okay, so this webinar will be divided in 3 parts. In the first one, we will try to compose models of genetic circuits from the Hill functions we obtained in the previous seminar. If you remember, we were working up from the central dogma, modeling that constitutive promoter, then inducible promoters, up to the Hill function that we used and found. So we will use those kinds of Hill functions to compose models for more interesting and bigger circuits. Then in the second part, we will see how can we relate these models and the parameters in these models with experimental data. We will talk about a bit about that. And then in the last section, I will show you an example, which is the incoherent feed-forward loop circuit, and also how to model that a bit and how to relate that with data, and how to, at the end, how to learn why the models are useful and what can we learn from them. Okay. As usual, Jake is taking the Q&A section. So if you have any question during the webinar, please ask it there, and Jake will try to answer it, and me at the end of each section we will try to answer those questions. Okay, but leave no question unasked, because if you don't understand anything, please ask, and so we can make this more dynamic. Okay, and last but not least, as usual, the videos of these 3 sections will be recorded, and then you will be able to access them from the iGEM webpage. Okay. So remember if you saw the previous videos of the last week. If you didn't, I recommend you to go back and look at that. We were introducing how to model synthetic circuits for synthetic biology, and we showed how we start from the schematic, then we make the biochemical reactions that represent that circuit, and then we get a model, and then we showed how we can get reduced models that are more easy to handle, and also they are closer to the data, as we will see today. Okay, but so now we want to model a genetic circuit, but imagine that so the first question you need to ask is, what do you want to do? And if you have a project in mind, there are other webinars related to how to deal with that. You may have an idea and you want to build a genetic circuit to address that problem or whatever. So what do we want to do? This is what I'm presenting you here is the famous, let's say, paradigm that any intelligent element needs to have, which is the sense, compute, act paradigm, that says that for you to make an action, to do something, first you need to sense the environment, the first block in yellow here. Then you need to compute based on that information, which is the think part, let's say. You get the information from the environment, and then you take some decision coming from this information, and then you make an action based on the result of the compute that you did before. For example, a robot senses the environment, it sees if there is an object in front of him, and it decides if it has to move to the left, to the right, and then it moves. So this is the three elements that we like to have in a system to make some intelligent decision. And in the context of synthetic biology and gene circuits, we can, the sense part, it can be a biosensor to measure a small molecule, to measure, for example, whatever from the environment, temperature, pH. There are many sensors which are in general encoded in promoters. So you can go to the part registry and look there. There are many promoters that you can use to sense different elements from the environment. Then from that information that you get from there, we want to compute to make to take a decision, and in general that's implemented as logic. For example, an inverter to invert the information that we got. Memory, like the toggle switch, detect levels, etc. There are many computations that you can do. And then the action can be the one that we would use here to change the expression level of a reporter, but it can be to express an enzyme or to express a small molecule that can be a signal to start the downstream process, or it can be a therapeutic. Any way that will lead to the action to what you want to do. Okay, so first you sense the environment, then you compute and make a decision based on that information, and then you act on the environment or whatever is the object of your thing. So as an example, we will use a sense compute act genetic circuit which detects arabinose. It has an inverter which is implemented with the TETR promoter, and then we express a fluorescent protein as an action depending on the calculation, the compute that we did with this inversion. Okay, so we will use this as an example to show the concepts of how to use the Hill functions and how to model this in a simple way. Okay. Okay. So this, I told you the circuit. Now let's see the implementation of this circuit in biological parts. We have in yellow, if you see the colors will be following up in the whole presentation. The yellow, it will be the sense part, blue is the compute, and red is the act. So how this circuit works, we have arabinose coming from outside that we will put into the experiment, and is the thing that we want to know how much there is, what we want to sense. Then we have the PBAT promoter, which needs the transcription factor RC to be present, which is in this case is constitutively expressed. And so when arabinose comes, it binds to the RC protein and induces the expression of the PBAT promoter. So up to here, we will have the sense part, and whatever we connect here, which in this case is the TR protein, but it can be anything else in whatever you want to build. So the level of the protein you put here will be expressed depending on how much arabinose you put into the medium. That's the sense part. Then the compute part, what it's doing is based on the PTET promoter, which is a repressor, and depending on the level of expression of this protein TETR, which is the transcription factor, the more you put of TETR, the less the expression of the protein you put here you will have. It's a repressor. If there is no TETR, it will be high expression. If there is a lot of TETR, there will be very small amount of expression. And then the last one, simple in this case, is just a fluorescent protein that we put here and that we will measure later on to represent our action. Okay. Okay, so now let's try to make a model of the different elements that we have here, the sense, the compute, and the act, and then we will connect them together, and we will get something interesting from the model. Okay. Okay, so here, what I'm representing with this Hill function, that it looks little bit big at the beginning, but once you understand the meaning of the elements, it's always the same. This one, someone, I don't know if he's here today, but someone in the previous webinar when we were talking about this, asked about why we are writing in this case, this is an activator, the promoter PBAD. So this is called the basal expression. If there is no RC, if you put the 0 here, sorry, arabinos, if you put the 0 here and here, all this term is 0. So what it remains here is what is called the basal expression, is what is expressed even when you don't put any, when the amount of inducer of arabinos in this case is 0. So that is what is called basal expression. If we write the Hill function like this, here we put the basal expression, and here we put 1 minus the basal expression. This is a way to write the Hill function that allow us to put here the maximum expression level. Imagine if this is completely induced, then altogether this will be a 1, and this will be the maximum value. At the end of this slide, I will not show you today, but if you want to take a look, there is the relationship between the different ways of writing the Hill function and a deeper explanation of what I just said. Okay, so then we have the inducer, which in this case is arabinos. The concentration of arabinos goes here, the Hill function, Na, and the dissociation constant of the inducer complex to the promoter, which is the Kd divided to D. And then here we have collected all the parameters from, let's see what we have there. In the alpha, we have the transcription rate of the PBAD promoter, which is K1. We have the copy number of this plasmid, where this construct is put in. If we have more, a high copy number, there will be a lot of expression. If we have a lower copy number, we will have less. Then we have K2, which represents the RBS, the translation rate, which is encoded by, in general, by the RBS strength. And then the decay rates of the protein that we put in the output. If you look this, okay, I went further. I wanted to show this. These two depend on the output, what you connect here, and the other elements of the Hill function and parameters depend on the module itself, the sense module, which are these guys. These ones I already mentioned. And the KD of the PBAD depends on many things. One model that we are using here, which is taken from this paper that I'm citing right there, which takes into account not normally the people takes into account the dissociation constants to between the transcription factor, and then between the transcription factor and the promoter, which are these two guys here, and the amount of transcription factor of the protein that you have, which in this case is RC here. But in general, people doesn't take into account much the copy number of the plasmid where this promoter is put in, the PBAD. But why is this important? Because it's not the same having 2 molecules of this when the copy plasmid is 2, then we will have a high probability that those 2 molecules are bound here, depending on the affinities. But if we have 700 plasmids and we have 2 molecules, then we need much more molecules to reach an occupancy level corresponding to the one we had before. So this is taking into account that information. If you have higher copy number plasmid, the KD that is representing this promoter will be much higher, because what it means you need a higher amount of the inducer of this guy to be able to reach the activation level that you want, okay? And that's why copy number is here also. So it will change, of course, the output, the level of the expression, which is given by alpha, but it also changes the KD, which is, if you remember, is the one that makes the transition of the Hill function from the low level to the high level in the case of an activator, okay? And this I already said before. So let's make a simulation and put some parameters there. Later, we will see how I kind of chose these guys, these numbers. But for example, you can go to the bionumbers page, which I forgot to put the link here. But if you Google bionumbers, I will put the link in the repository. I think I did it already for the previous one. And you Google parameters for the translation rate in E. coli, for example, and then you will get some examples of numbers taken from the literature. So, for example, in this case, this guy, if you remember, is the percentage of the total expression that we will get when there is no inducer, is the basal expression, which it will tell you this level up here. The KD, which it tells you the half concentration, which is the place where the fold change is half, which is around here in this case, around here. 400 micromolar of arabinose, and then the alpha over theta. In this case, I select a whole number for these two guys. Then you will have to, depending on the protein that you put there, this may change. If it's a stable proteins like the ones we generally use, then that will be mainly given by the dilution rate of the culture, the growth rate, the related dilution. And that number will change depending on how the culture grows. But for now, I will put to all these one number, which is, for example, 710 to the 4 molecules. If we do this, then we get this kind of kill function, which tells us, for example, that if we put arabinose less than 10 to the 0, which is 1, if we put less than 1 micromolar, we will get no change in the amount of this protein. In the expression of this protein. If we increase the amount of arabinose that we put, the expression will be going up until this curve goes on. And if we go higher than 10 to the 3 micromolar, which is a lot, 1,000 micromolar, sorry, it will reach also the plateau here, the saturation, and it will not change. And then we have a useful range if we want to sense. If you remember, this is the sense part, so we will be able to sense concentrations changing from 10 to the 0 to a little bit lower than 10 to the 3. Okay? So this is an example of the simulation, what the models give us, this equation, for a set of parameters which represent, in the future, they will represent the construct, okay? So, but what happens if we change for example, the RBS that we put here, this guy? We have different RBSs. If you recognize, these numbers are from the PARP registry, and these are the most commonly used RBS ever, like thousands of times each one. And so what happens if we put these three? I chose these three because it's the ones that I use in my lab. But this one is a very strong RBS. This one is a middle one, middle high, and this is a low one. So I want to know what happens if I change this, the RBSs, and how I have to change the parameters to represent this change in the RBSs. So what is the effect of making these changes in the Hill function? So we take a look at the equation. If you remember, the RBS will make a change, will have an influence on the translation rate, which is given by K2. If you have questions about this, please go to see the webinars from the last week, where we did the model and we got all this from the central dogma and from the model of the constitutive and then the inducible promoters in detail. And so if we change the RBS, we will be changing this, and alpha p-val will be changing, and we will change this. So if we look here, the only thing that will change this, meaning that the curve that we saw in the previous graph will go up and down depending if the RBS is a strong one or a middle or a weak one. Indeed, if we make the simulations and show that, we see that the one that we had before, the B30, is the higher one, the one which has a stronger expression. and then the other two, the curve is only moving down, meaning there is no change in the KD, in the sensibility of the p-valve promoter is sensing the same range than before, but we are having a higher expression in the output in theta or a lower, depending on the RBS. Okay, and these are the values for the. If you go to the registry, you will see some characterization of the RBSs, which is given as a relative. So in this example, I use the relative, the 30 as a 1, and then these ones, I change the values relating to that relative scale between the RBS. That's a way of doing it. Okay, so we have that. What happens now if we put this construct in different plasmids that have different plasmid copy number, depending on the origin of replication? If we do that, what we will have, for example, we put that in a high copy plasmid, 300, like the PSB1C3, for example, the one from the registry. If we put that there, 300 or more, we have what we had before, the full line curves. I took the one from the middle for visibility. If we put the curve into, sorry, if we put the construct in a lower copy plasmid, a medium copy plasmid with 50, for example, then the curves go down, but they also move to the left. That means we are also changing the KD of the promoter, okay? So that's something to take into account, because in general, people think that changing the promoter only changes the level of expression, but it also changes the KD. There is a question in the Q&A. Someone anonymous says, would there be a situation where you would want to use a less sensitive RBS? Yes, definitively. We will see in the end of this section. I will show you why you may want to have different levels of expression. Because when you connect different systems, they have to be compatible. The input of, in this case, remember we are connecting the sense part of the circuit, the sense module, let's say, with the compute, and then with the act. If the output of the sense model is not compatible with the input of the compute module, then this will not work. So actually, this is what we want to show here, and that will answer your question, okay? So let's move on, because we are taking too much time. So as to show you the numbers of what just said from the proposed original numbers, from the high copy number, we had this for the B07 to the 10 to the 4, and the K-val was 440 or something like that. If you go to the medium copy plasmid, this goes down, of course, because we are having less expression, but also the KD goes down. We need less inducer to have the same response. If you look, this curve, the half concentration is around here, and in this curve is around here. So it moved to lower concentrations of arabicus, okay? Yes, I will show. So there is another question. It says, are there many cases where the modeling is not actually modeling what is happening in real life, and you have no idea of why? Yes, many times. But we will try to deal with that and to make the model answer the questions that you have. But one important thing is that you need to be aware of what you want to get from the model, because sometimes what it happens is, oh, the model is not telling me what I wanted to tell me. But sometimes that happens because I'm not asking the right question. So always first ask the question, what do I want to get from the model? Why I want to model this and not something else from my system? And then maybe the chances that you have something like what you were asking are low, okay? Okay, let's go on. We have a few more. So now let's focus on the compute module that we have. if you look, it's another Hill function that represents the promoter. But in this case, the difference with the one before is that here in the numerator of the Hill function, we have the KD instead of the concentration of the inducer. This is because this one is a repressor, okay? When you have KD and KD here, it's a repressor. When you have the inducer down in the denominator and in the numerator, then it's an activator. And how to look at that? Put a 0 here and see what this number is. The maximum number that you will get from this expression is when you put a 0 here, meaning that this is a repressor. There is another question related to the copy number, and it says, is there an absolute threshold at which medium copy becomes a high copy? No, that's how people in general classify the plasmids, because it's not a continuum, it depends on the origin of replication. So there are some origins that have normally higher copy than another ones. For example, the PMB1 is a very high copy number origin, and the P15A, it has like 10 copies. So that's a low one, and the other one is a high one. But there is, it's just a way of describing qualitatively the amount of copies you have. Okay, so we have this. If you look, it has the same shape. Everything is similar than before. And this one I go quicker because we already did it before. I'm doing the same exercise done previously. Different RBSs, in red the strong one, in orange the weaker RBS, the B0032, and then we have high copy plasmid, the full line, and medium copy plasmid copy number in the dash line. And again, this one is more clear. When you change the RBS, it goes down from here to here, maintains the KD, doesn't change. But if you go from a high copy to a lower copy plasmid, you change the level in the y-axis, but you also move the curve to the left, meaning that the range of the input, which in this case is theta, it's a different range. Here I am acting in a range from, let's say in this one, from 10 to the 2 to 10 to the 3-ish, a bit more. And in this one, I'm going from 10 to the 3 to 10 to the 4, and so on. So it's a drastic change, because here I'm in the thousands and here I'm in the tens of thousands. So it's important to take that into account. Okay. So now let's put it together and make the model of our sense compute act device. If we put them together, basically what we have is that we have simultaneously the two equations that we had before. And so let's make an exercise that will try to answer the question that we had at the beginning, if I want to have a weaker RBS. So imagine that we have these 4 constructs in mind to make the construct in the lab, because we have availability having the different RBSs and the different plasmid copy numbers, depending on where we clone this part, okay? So, and then we have a useful range of arabinos that we can pipette, let's say, or that we decide that we want to be able to sense in that interval from 1 micromolar to 100 micromolar, okay? And imagine that we say, okay, let's say I would like to see what happens if I make the construct of the one on top, the higher copy plasmid and the stronger RBS. So that means that we will hit that curve with our initial range, and that will give us a range of output theta for this module, which is in this range, okay? And then this will be the input of my second module, if you remember, is the input of the inverter, which is the theta promoter. What happens if we make this one into the input of our second module? And imagine that we want to use the red, this one, the one with the arrow, which was the low copy mid copy, lower RBS, high RBS, sorry, the B30. Then our range of input in this case will hit this curve, and the output that we will get will be in this range. So what happens here? What we got from this analysis? Look at the input range of arabinos and look how our output RFP is changing when we have those changes in the concentration of arabinos. It's almost undetectable, almost insensible to that, because of the way we combined the RBSs and the copies of the plasmids. Okay, so yes, it means that in the answer for the previous question, sometimes we need to have a weaker expression or a higher expression to make sense in the total construction of our device. Okay, for example, in this case, one may want to use, for example, this curve to have a higher range here, or to use this orange instead of this one. So if you compare this one and this one was with the B30 RBS and the medium copy number plasmid, and this one is with the strong RBS. Sorry, this is with the low RBS, the B32, and with the I got messy here. The idea is that in this case, instead of using this one, you will like to use maybe this orange one, which will have a much higher range in the output responding to the initial change in the input. Okay? Okay, so this will be all for this part. Thank you for listening. And if you have any question, you can ask them now, or you can ask them on the way in the next parts of in the next two sections, sorry. So I don't see much questions. We were answering them along the way. So we will start the second one.